Well, good afternoon, you all. We're back. Welcome back to our second round of presentations. We have two lectures in this afternoon. And just to remember, you can make any questions in the chat, and we will try to answer as many as possible after the talks. So the first talk in this afternoon, we have Professor Manuel and Melo talking about using MDAnalysis for structural work before and after simulation. So we are very happy to have Professor Manuel and Melo here, and I'll give a brief introduction about him. So he leads the multi-scale modeling lab of the Universidade Nova de Lisboa, which focuses on molecular level details of biomolecular interactions. To this end, they employ coarse-grained, atomistic, and hybrid MD simulations. He is NWO Veni and Marie Curie Fellow at the Molecular Dynamics Group of the University of Groningen. He has an H-index of 27 with more than 4,100 citations. So, Professor Melo, thank you so much again for your availability, and you may start whenever you're ready.
Thank you so much, Rodolfo. Thanks a lot for the invitation. I'd also like to thank Luciano, who was the one who reached out to me and made this invitation. I just want to clear something. I'm no longer affiliated with the University of Groningen. Okay, that was during my postdoc time. That's when I was a fellow there. So I've been independent for quite some years now. But onto the topic at hand. So using MDAnalysis. So some of you might have heard of it. Some might never have heard of it. The point is, when Luciano asked me to present here, I was a bit intrigued because I don't really do a lot of research into material science. But then it became clear, okay, no, he was interested in my expertise or in my background working with MDAnalysis and developing MDAnalysis as a tool. So that's what I'm going to talk about. So my examples are going to come more from the biological sides, from proteins and membranes and stuff, but it doesn't really matter. This is just to showcase the tool. Also, to make things clear, ever since having my own group, the time to dedicate to developing open source software such as MDAnalysis gets critically, it just disappears. So I recently became an emeritus MDAnalysis core developer, which is a very nice name for someone that is no longer expected to actually have to do the day-to-day managing and the voting in the day-to-day decisions. But I'm still very much involved I like to be involved into the code development, and that's still pretty much open. So just so that it's clear, my position, my place in the structure. But without further ado, let's see. Okay, what is MDAnalysis? What can it do? Why should we use it? Okay, so just copying from the GitHub description, there you have it. It's a Python library. Python is all the rage now. It's very nice. I mean, I'm all for it. Maybe other languages are going to be better in the future, but right now Python is very big, so it's great to have a library that works in Python. It's focused on many-body systems at the molecular scale. So essentially this means molecular dynamics, or at least anything where you have structure and coordinates, and possibly those coordinates change over time. Okay, so that's the very, very simple aspect of it. Written by scientists for scientists, it's always good to know. Birds of the feather tend to have the same problems. So, and one of us writes something to scratch their own itch, it might scratch other people's itches as well. So that everyone gains from there. But this is the official description. Why do I think it's important to use is, ever since I started working in molecular modeling, in structural modeling, structural analysis, I realized that there are very few the cases where you have very, very, very well-defined analysis in a way that you can readily use ready-made tools. So you often have the problem that you have your simulation, you want to probe it for your hypothesis. So you have, if you have a simulation, you usually have a lot more information than you know what to do with, and you have a question, And either no one has written a tool to answer that question, or if they did in a way that it might need some tweaking and some customization. And a lot of times, especially the analysis and the questions that are not so complex, I find it much more rewarding and much better tuned to actually write the code myself or write and tune it to my question than to actually shape the question to try and fit existing tools. So I think that is, for me, this is the ultimate, the most important aspect why I like this attitude, because it makes it very easy to make my own analysis tools, essentially. Okay, how to get it? Now let's go to the practicals. It's distributed with conduct because it makes sense, scientific stuff. But you can also get it from PyPy with pip install. You have some options if you want to bring in more dependencies so that some of the more esoteric analysis run out of the box. But essentially, this is a Python package, and being a Python package is a nice object-oriented package. It has this object-oriented interface, and one of the core objects here is called the universe. The universe is what you call the system, let's say. Okay, it has two main aspects: the trajectory and the topology. The trajectory is where we hold the time-dependent system data. So there will be the positions. Imagine now that you have an MD simulation. Over time, you'll have simulation frames that you've written, so you have the positions. Possibly you'll have velocities or forces. If you have a varying box, the box will change over time. Maybe there's no box, but if there's a box, it can change over time. Even the time itself is something it's a variable that you can hold. And the very nice thing about this MDAnalysis package is that it exposes this all as much as possible as NumPy arrays. So if you're used to NumPy, you know that this is very, very handy to do rather fast and handy vectorial computations. Okay, so you can imagine this being a stream of frames, each frame being for instance, for the positions being a two-dimensional array where you have your atoms. So let's say, I don't know how many, 20 atoms times X, Y, Z columns. So this would be how your data would look like, for instance. Okay, if it is only positions, then you could have more for the velocities and so. This also works for formats that are single frame. You can imagine a single structure PDB file. It's abstracted as being a one frame trajectory. So it's the same thing. And and what I meant to say here is that, contrary to some software, the entire trajectory is not loaded at once into memory. So this is quite lightweight, which is very welcome. Essentially, as you go, you load each new frame data page into memory and discard the old one. So this is very handy. If you do need to load it all, it's possible as well. So this gives you all the power. The topology holds typically the time invariant system data. So atom names, the masses, the charges I didn't put there, but also the hierarchy. So what is a molecule? What is a residue in that molecule? What are the atoms of each residue? And then the connectivity. So which atoms are actually bound to one another? Also the number of atoms, how many you have. So that is all part of the topology, and not all of these things have to be available all the time. Depends on the formats you have when you did your simulation and what you're reading into MDAnalysis. Now I'm putting here a couple of exclamation marks because, for instance, connectivity need not be constant during a simulation. There's reactive force fields, and there's simulations where you break and make bonds. And MDAnalysis right now won't be able to deal with it very cleanly. Even worse would be varying atom numbers. Okay, so if you're simulating, for example, in an ensemble where your n is changing, your number of particles, this will be tricky to represent properly with MDAnalysis unless you have some tricks. So this is something that right now is not possible. But it's also, these are relatively corner cases, so most of the things should be covered. This is a bit of a top-down look into this. There's also a bottom-up look into the interface, which is centered on the atom group object. So this is, as its name says, is a group of atoms, represents a group of atoms. And through this object, you'll be able to access, for instance, those atoms' names, their masses, charges, whatever, and at the same time, their positions, velocities. And so from this kind of object, you will be able to access the topology and the trajectory filtered by which atoms you actually put in the group. And we're going to see how that can be done. There are some hierarchical notions, so you can also talk about residues and residue groups, segments, and such. They tend to be a little bit less useful than the atom group. I myself use mostly atom groups and residue groups. That's essentially what that solves most of my problems. And also the single objects, the singular objects, single atoms, single residues, they can be useful sometimes, but you'll see that most of the times the atom group is what you need. Now, we talked about topology and trajectory. What formats are we talking about? So we're talking about 20 plus formats for topology right now that are coded into MDAnalysis, and trajectory, I think we're at 40 something. I haven't counted them exactly. This is pretty handy. This allows you to make tools that can be quite abstract. So you can make a tool that, as long as it gets a topology and a trajectory, it will work. So it's up to the user just to find whatever format that MDAnalysis takes in, and that should work. The abstraction is not perfect because not all topology formats and not all trajectory formats have the same attributes. For instance, there are some trajectory formats that only have positions, but there are some that have velocities or forces. So if your analysis depends on velocities or forces, of course, then you're going to have to need one of those formats. Same thing for the topology. Some topologies will have more information than others, like atom types, for instance, that gives information of how those things were simulated, the potentials acting on those atom types, also bonds and such, that need to be made compatible with your analysis. But overall, from a bird's eye view, this allows for a lot of abstraction and a lot of interoperativity, and hopefully the things you write won't bump so much into problems of needing attributes that topologies or trajectories don't have. So still on the object, going back to the object-oriented interface, and so the organization is that the universe has a collection of atoms. So this is all the atoms in the universe. And these atoms are organized into residues, which in turn are organized into segments. This is a bit a historical consequence of how MDAnalysis came to be, in that this represents roughly the organization, for instance, in the PDB file, where you have atoms that belong to residues. An atom cannot belong to two residues, so an atom belongs only to a residue. A residue can have multiple atoms. And then the residues belong to chains, which would be in this case the segments. Yeah, the segments then are the concept more or less for molecules, although this is not always a one-to-one identity. It really depends on the format of the topology that is going in. So that's why I end up usually using mostly atoms and residues. Depends on the format. For instance, I use mostly GROMACS, the GROMACS software, and for GROMACS, the output files, the output formats, they don't really have what is a molecule. They don't write that down, especially in structural files. So it's essentially residues end up being a way of distinguishing what is one molecule and another molecule, because and depending, of course, if you have X residues per molecule. For instance, I work a lot with coarse-grained modeling, and in my modeling, for instance, when I simulate a membrane, one lipid molecule will be represented by GROMACS as one single residue. So this is something I end up using. I don't use segments that much because of that. So some code examples. So I'll show quite some examples over this presentation, and I hope to make it kind of simple and concise. That's the whole idea of Python and this kind of packages. And of course, scripts can become quite complex quite very quickly, but hopefully I'll exemplify how things can be streamlined. So we usually import MDAnalysis abbreviated as MDA because no one likes to type. For instance, here I'm loading a PDB file, and we load it just like this. It's very straightforward. You might be asking, oh, but you said that the universe needs a topology and a trajectory. There's only one input here, so how is this going to work? The thing is that a PDB file can be its own topology and its own trajectory because it has its topological information, the atom types, the masses, etc. Not the masses, but the names, and the masses can be inferred. And at the same time, the coordinate, the one-frame trajectory at the same time. So this works. So it's a two-in-one loading, and then you can access the atoms. And this particular PDB, which is of a spike protein bound to antibodies, because that's all the rage now, of course, has these many atoms. Then you can also inquire about the residues. As I said, okay, a single load for both aspects, and we can now inquire about the positions of the atoms, for instance. So this is what I mentioned, that this is made available to us as a NumPy array. Okay, and this seems very simple, but herein lies a lot of the power of this library. Because if you know your array around NumPy, you already see a lot of things that can be very quickly done, very easily done, just by having this information at your fingertips so directly. Okay, there is no topology object directly accessible, but there is a trajectory object. So, and in this case, it's a single frame trajectory object. It's not very useful on its own, but I'll show an example where it becomes a bit more obvious what can be done with it. Then, okay, we just saw an example where you had 25,000 atoms. Most of the times you don't want to work with all of them at the same time. Maybe some of them, like solvent, you probably want to ignore most of the time anyway. Which means that you need to select some of them. And the most straightforward, and if you're used to Python, this will feel very natural, is to use slicing or fancy indexing if you're also used to NumPy. What this means is that you can essentially index the atoms object of this universe, and essentially you do a subselection there. So in this case, I'm selecting from the 11th till the 100th atoms. Okay, then you get 90 atoms. This is nice. This is very straightforward. It requires you to know where they are in the sequence, where the atoms are in the sequence of your topology. In 25,000 atoms, I mean, if you know what lies in the position 10 to 100, it's fine. But it can be a bit not so easy. We can use fancy indexing. As it would be used in NumPy, in that instead of using ranges, you can just specify exactly I want the 1st, the 6th, the 31st, and the 101st. So this works also very well. You get an atom group with these 4 atoms. And if you have a compatible mask, you can also use a Boolean mask, in the sense that if you had an array of 25,783 trues and falses, you could use that to index the atoms object, and you end up with a subselection of only those positions where that mask were true. Okay, but as I said, this mostly involves you knowing what is where. But how about combining what you know about the topology to try and do some selections? And that's where this nice method comes in, the select atoms. So here you'll be able to start using syntax, as for instance you might have used in VMD or other software such as that, to try and get the atoms you want. For instance, you can just go to the universe and say, I want to select atoms that have that correspond to this search string. And this search string there's some allowed keywords, quite many actually, and it's very easily this one. We're just picking all the atoms with name CA, so we were selecting the alpha carbons of the protein in this case. And now we're down to much more manageable list of atoms, and one that makes sense. So if we want to work with the backbone of a protein, often the alpha carbons are a very good simplification of it. And this is a very common first step in a lot of analysis. We can also go for the residue names. You can combine stuff with logic. So I want all the methionines that have all the atoms that are that are part of methionine, and they have a name CB. So here we have. Okay. Again, I'm exemplifying with proteins, but this should be is readily extendable to other cases. So time. Actually, stuff. Let's look at the root mean square fluctuation. This is essentially the root mean square of the difference over the entire trajectory minus its average. Naively, this needs to be done in two passes: first to get the averages, then to get the differences. But how would this be done? And this is an example that we had in the orange are the parts that are MDAnalysis, and in blue the parts that is actually NumPy action. Very simply, in this case, you're loading a universe from a GROMACS topology and a GROMACS trajectory format. Very good. You select some atoms. Now these ones that correspond to the alpha carbons. Fine. The example I just showed. Now here, this means that we're doing NumPy stuff. We just prime some variables, some arrays. But the important thing comes here. This is essentially a for loop, and what we do is we're going to iterate over this trajectory object. Okay. And the interesting thing here is that what comes out of this object is this is a trajectory. We get what's called time steps, this variable, but we are actually not really using this variable anywhere. What happens here is that in MDAnalysis, and this is important, is moving the trajectory automatically moves what every atom group is pointing to. So this atom group is pointing to the atoms that correspond to the alpha carbons, fine, and they have some positions in the first frame. So after you load, you're by default on the first frame, and if you ask the positions of these atoms, that's what you'd get. But when you move over the trajectory, this selection still refers to the same atoms, but now their positions are going to be updated. I'm going to show an example later a bit clearly, but essentially doing this iteration, we can do this computation. This is the algorithm of the single pass. Essentially, we just go and get the positions of these and work with them in the means, and at the end, we just do the square root of it all. And finally, we can plot it using matplotlib. We can also, for instance, get the res IDs from the alpha carbons, just use as the x-axis, and you get something like this. So this is a very quick example. The math might be a little bit slightly dense because it's a fancy algorithm, but the important thing here was the statefulness of this universe object. Especially the trajectory part. So contrary to what I've seen a lot of people expect, you don't need to specifically tell an atom group, look, go and fetch the positions for the current frame. No, by the moment you move to the current frame, all the atom groups are now pointing towards that, to that. Okay, so this is a very good aspect of. It simplifies a lot, but you have to wrap your head around this philosophy. So still selections. Okay. Now we can. There's more complex stuff that can be done. So not just looking at names, you can do geometric stuff. Okay. So I can look at lipids, a lipid called POPC. This is a system that had lipids and a protein, a membrane with a protein. So I can ask for all the POPCs, all the atoms of POPC that are within 10 angstrom of any protein. Okay, this works, and here we have it. Okay, I'll call this near, and they have this. It has these positions. Again, this example of separate loading of topology and trajectory, and like the first example. Also another caveat, so this selects atom, not necessarily entire molecules. So I can just be chopping a lipid in half because part of it was within the cutoff and the other part not, okay? Now, to exemplify what I just said, if I now move to frame 11, and this is, well, the 11th frame, frame 10, the 11th frame. It's Python, you know. I would do like this. I essentially just get that element, the element 10. I get this feedback. So apparently, I didn't change anything. I didn't modify anything of the atom group. So the atom group is still the same. It still corresponds to these 234 atoms. But if I now ask for the positions are changed. Okay, this is what I was explaining earlier. Okay, so changing the frame implicitly changes the loaded atoms. Okay, so if I wanted to get now the closest atoms in this frame, now I'm in the frame 10. If I want again to get the closest atoms to the protein, I need to rerun the selection command. What this means is probably these lipids, very likely, they will have diffused away a little bit. That's what usually happens. And well, I'm having here lipids and lipid positions that might not be within that 10 angstrom cutoff I established. So if I just repeat that command, I will have that fixed. Okay, there is a bit of, it's not syntactic sugar, but it's some helpful construct, which is when running select atoms, we can specify that we want this to be updating. Okay, so then you get an updating atom group, and it's an atom group that responds to the changes in the frame. So here I'm not showing the positions. I'm showing just the number of atoms in this atom group. So I'm at this frame. I get 250 atoms with this criterion, and if now I move to frame 20, the same atom group without doing anything else now corresponds to a different number of atoms because it's automatically updating. This is a very brief overview of what can be done. There is many more selection criteria that are possible to use. For instance, something a little bit more complex. Let's say we have a membrane and we have some peptide on the top and bottom of that membrane, and I want to get the ones that are on the top of the membrane, but only around the lipid POPC, and I want them to get them as whole fragments. I don't want that effect of chopping the molecule in half because it's outside of the cutoff. So whatever has a foot inside the cutoff is all counted. So this would be the selection string. It gets a little bit complex. It's still intuitive and there's logic to it. Just to showcase that much more is possible than just the simple examples I have here. Okay, so it's in the name, MDAnalysis, right? So we are expected to work with analysis. And honestly, I find this it's not exactly a misnomer, but what I find the strongest point of the library is what's called this core, which is what I showed you, or what I've started to show, which the interface with the topology, the trajectory, all this NumPy exposure, the selections, the very powerful selections. There's no analysis here yet. The analysis I showed, actually, the example coded the analysis. There was no pre-made analysis. But in fact, there is an MDAnalysis.analysis submodule. So that's where some pre-made analysis live. As I said in the beginning, the moment you write an analysis, I mean, it's for a certain purpose. It might have some flexibility, but I'm sure someone is going to come around and want it to do something else. So, and my feeling with these analyses is a lot of them are a little bit like that, but the 20 plus that are included with the library are the ones that are the most generic, I'd say, or at least the ones that might be employed without so much need for modification. So you have PCA, root mean squared for the RMSF. We didn't need to code it. That was an example, but that's also there. Stuff like water dynamics, H1 analysis, a lot of it, contact analysis for the people that study membranes like myself. There's stuff that to study leaflet behavior. What is one leaflet? What is another? So 20 plus of this, and they all make sense from this standpoint that I said that, okay, I don't find them redundant. I don't find that I need to rewrite them every time I use them. So I think this makes sense. But in the future, MDAnalysis is moving towards the kit solutions, similar to scikits, in which, especially as more and more complex analyses are being written and developed, instead of covering them or including them alongside the main library, these will essentially be developed in parallel, and essentially by the people that actually developed them and that made the research that led to their development. That's very good for the core developers, because it releases them from having to field bugs in analysis that might not have to do with their own work, so which would be very hard to debug. So that is a very, I think it's a very good plan. I think it's going to be, as just like SciPy did the scikits, I think this is going to be very successful. Okay, but for the analysis that exists, what can you expect and how would they work? This depends a little bit on each analysis. The documentation is there, so and they're all rather simple to use, most of them. But for instance, let's say we want to compute the RMSD, so the root mean square deviation of a set of atoms. In this case, I'll be looking at the coarse-grained protein and looking at their coarse-grained backbone atoms. It doesn't really matter what it is, but the example here, for instance, we import this analysis module, the RMS. We load again this universe with a trajectory, and I select these backbone atoms. In my coarse-grained model, I use the Martini model. They're called BB. Okay, so very simple. There, done. And then this particular analysis, all I need to give to it is the backbones, or the atoms I want to compute the RMSD of. I could optionally give it a reference, but in absence of a reference, it will just say, just assume that the current frame is the reference configuration to compute the RMSD against. And then I have this option to run the analysis. This is where the magic happens, where stuff actually gets done. And this also gives me the option to have a start and a stop parameters and a step parameter. So if I don't want to run over the entire simulation, in this case, I just set a step of 100, so to be quick. This was a very long simulation with around 50,000 frames. Then only analyzed 500, and after it's done, this analysis object is modified internally, and now it has this RMSD attribute. And this RMSD attribute is this 2D array. What you have here is the frame number, 0, 1, 2, so on. The 0, 100, 200, because we have the steps of every 100 steps. The time corresponds to that. So if the trajectory has time information, you'll get that. And here, the RMSD we're looking at. And as far as RMSD goes, you can see that the one, well, it should be 0. It's only not 0 because of numerical precision, is the one corresponding to the first frame, because it's the same as the reference. All the other ones, they're higher, not that high, but 1 point something angstrom. And that's it. Okay, here it is, the shape of this array, showing that we have almost 500 frames, because we have almost 550,000 frames total divided by 100, and these 3 aspects. Okay, so this is one typical analysis, and you see you can get something very meaningful with a handful of lines. Right here we have 1, 2, 3, 7 lines, 6 lines, and you have something important. So this is quite simple, and it's a good aspect of Python and of this module. Okay. In the MDAnalysis also makes it quite easy for you to write your own analysis. Okay, and that is quite important for a number of reasons. It really promotes encapsulation and modularization. Of course, if you're just doing a quick hack to get a value to complement some other analysis, I mean, don't do this, don't go this route. But if you're going to do something that you yourself might use down the line more frequently, and especially your colleagues, and at some point you'll probably want to put this in the repository, other people start using it, this is really this really becomes handy. It really makes it easier, this modularization. So you'll basically create a class, an analysis class, that will take care of all that. And what is all that? So just by creating a class that imports from this analysis-based object, and I'm talking now about Python concepts like inheritance. So yeah, you'll need to know a little bit of Python to go into this direction. A lot of the boilerplate is dealt with already. So just like I was able to use the run method and provided step of 100, just by importing from analysis base and defining your analysis within this context, automatically your analysis would have the same thing, the same possibility of starting at a given frame, ending another frame, and having a step of such. So all this. This also exposes you to future enhancements. If the analysis base gets an upgrade, let's say if the parallelism gets added neatly to MDAnalysis, then your analysis could also automatically become parallel. Okay, I don't think it's going to be that simple, but this is sort of an example of what an upgrade of the base could mean for your analysis. And what I have here is the skeleton of the analysis base class. There's these 4 methods that you can change, but essentially when you instantiate your class, when you create this, let's say when you created the RMSD object, this happens. So it runs the init method and the prepare method. This is, for instance, good to regarding your analysis, to prepare, for instance, counters and accumulators and stuff like that, and also to parse in whatever the user gives as options. In the RMSD example, it was the backbone group that needs to be stored somewhere. That's it. And then the most important part here is this. This is what is going to be done every single frame. So every single frame, this function is going to be called. This method is going to be called, and it can do a lot of things. It really depends on your analysis. And finally, optionally, there is a conclude step. So especially if you're doing average or something, you're at the last step, you might want to actually average what you accumulated and such. So this is just a skeleton, but let's look at an example. What if you wanted to compute the end-to-end distance of a soup of polymers? Okay. What would we need to know? Well, we just need to know actually what are all the starting atoms or all the ending atoms. So our initialization method would look like this. The user would just provide what are the starts and the ends. Of course, these would have to match. If there's 100 here, there should be 100 ends there. And we just store that together with the object. Because this is an inherited class, then we would need to actually instantiate or set up the base class as well. And then, because we're going to, in this case, in this analysis, this example, this is going to do the average total end-to-end distance. So we're going to be doing a sum over every frame. So let's start here the accumulators, the total end-to-end, and the number of frames, because then we're going to divide one by the other. And here is where the actual juice is. So we have this single frame method. So every frame, what are we going to do? We're going to use this method that comes also with MDAnalysis, which is calc_bonds. Essentially, we're computing the distance between the two atoms, the two sets of atoms, 1 to 1. And what it takes is, again, the positions of one, the positions of another, and it also needs to know what is the box size. Okay, and the box size is important because you can imagine in simulation the box can change, and the algorithm needs to know if a bond goes one way or the other way across the periodic boundary conditions. Then essentially we just get all the distances, we sum them all, and we add them to this total. We say, okay, one more frame went by, and this will be executed all the frames, so over all the frames, and finally we just need to compute the average, which is actually just divide by the total number of frames. So this accumulator total divide by the total number of frames. And because we might have more than just one polymer strand, we should divide by the number of polymer strands, which is given in this case by how many atoms we actually have as the starts. For instance, if we had 100 strands, we should have 100 starts. Then we have the average per frame, per strand, and we can put it in this attribute, this results attribute. And this is how an analysis would look like. This is, I mean, it's a simple analysis, but it's already a quite useful one. And it took very, very few lines. And by using the analysis base, it already gets quite powerful. As an example for this, then, okay, we define the starts and the ends using appropriate selection strings. Depending on your molecule, of course, what your polymer is named, what each end is named as, or what is the criterion you use to define that. Then you create an instantiation of your end-to-end distance class, and you have to pass to it these two groups that will become the AG1 and AG2. We just use the run. In this case, I'd say, oh, I wanted to skip the first 10 frames because I want to consider those as being equilibration. Okay, so we do the run. This object gets modified in place by the run because now it should have this results and E2E distance that we can print. Okay, so this becomes very, very simple to use in the future. Of course, you have to pay a little bit of price in developing it. It takes longer to develop initially to do it cleanly, but then it pays off in how simple it can be used. All right. So already showed a bit of core functionality that is central to all this, and one of the most useful core functionality has to do with distances. Right, I showed something in last slide that was calc bonds, but essentially this all builds on top of this function here. It's called distance array, and this distance array works like this. Let's say if you have a system that has two protons. The first one is the first 1,000 atoms. The second is the second 1,000 atoms. Let's say you want the all-to-all distance matrix of all these 1,000 versus 1,000 atoms. Okay. What you're going to do is simply use this distance array function, give the positions of one of them, positions of the other one. You have to give the box again if you want to take periodicity into account. And what you get very simply is this NumPy array of 1,000 by 1,000 with the distances already computed for you. Okay, so this again seems super simple, but as you can imagine, probably if you already had to do some sort of analysis yourself, you very likely had to compute some distances, and this is a very handy way to do it. Why do we actually use a function? Because we have access to the position, so using NumPy, it's also one or two lines, and we can get exactly the same array here. Well, this relies on sped up distance calculation routines. So this can be much more advantageous over using the pre-made NumPy stuff. NumPy is already pretty quick, but this can speed up more stuff. And this is especially so when computing contacts. Okay, so contacts can make use of tricks like cutoffs and boxing and chopping the boxes into cells of less than the cutoff, a bit just like the MD software does to speed up. So this is one of the reasons why using the functions in this submodule and this is very, very important. But there's other stuff. There's other measures. You can measure angles, can measure torsions, can measure centers of mass, centers of geometry, radius of gyration, stuff like sphericity, which is more exoteric. There's a lot already implemented. So, and this makes the core of most analysis that you'll need. Then there's also another aspect, parallelization, and this is very dear to me. I actually spent quite a lot working on this. Why do you want to parallelize? Essentially, it's speed, right? And sometimes it feels like you don't need to, because sometimes you have simulations that go for weeks or months. So what if your analysis takes 6 hours instead of half an hour? Come on. But yeah, it is important. And not only that, in a daily workflow, if something takes 5 or 10 minutes, it's one thing. But if it takes 1.5 hours, it's already a very, at least for me, it's a very big break in my productivity. And this is exactly the kind of speed ups that we can be looking at, because nowadays machines with 8 cores, 16 cores are quite common. So if you could leverage those cores just to speed up stuff by 8 or 16 times the factor of 16, that already really makes this difference. It's something that, okay, it's just you just go for a coffee and it's done, or something that you might as well just start doing something else and come back later. So yeah. So how are we going to parallelize? And the most natural here is to do it over frames. And I'm going to show exactly how I mean this. But essentially, most MD analysis, they occur over frames. So you're going to iterate over frames. And most of those involve calculations that are independent from other frames. And I'm saying this, okay, it's addition needed because most have a lot of, a lot of caveat there. But in reality, there are analyses that are not so readily parallelizable over frames, or you don't want them to do it over frames, or that the calculations that you do in one frame there have to be used in the next frame, and that makes this trickier. But since a lot of them are well behaved, let's go on with those. Because that allows for a generic implementation, okay? And typically this is also, if the computations are independent from other frames, you can get an almost embarrassingly parallel, meaning that you get a linear scaling compared relative to the number of processors that you have. So when you parallelize over frames, usually you can do it like this, you can do it blockwise, or you can do it interleaved, something like this. I'm trying to speed up. Let's see if I can finish this quick. This is usually the best one because it has a built-in load balancing. The only thing that it might require some sorting of results, because then each worker computes a result interspace. So when you get it back, you'll have to de-interleave them. There are several modes for parallelizing. You have the thread-based, but Python is not very good at that. I'm not going to now go into much more detail here. Python is better at process-based ones. Maybe it's going to change in the near future. But essentially, this involves either forking child processes or starting independent process and using stuff like MPI. This has an advantage, for instance, of working over different computers and communicating over the network. So this is how you can actually parallelize over more cores even than you have in your own computer. So I'm going to skip about these details of the different sorts of parallelizing. There are some implementations of parallelization. So there's a rather straightforward, it's not very hard to actually use pure MDAnalysis and using then this multiprocessing or MPI. We have an example in the last workshop we gave. There's a link along the description, but I can ask also the organizers to send out the link for the workshop. But if you Google it, you'll find the 2021 workshop. There is examples with this. There's also this PMDA, so parallel MDAnalysis, that implements a parallel analysis base class. So if you make your analysis inherit this base, then you get already automated parallelization. And this is the one I developed. This implements a per frame parallelization that also can use MPI or local parallelization and some command line convenience. So this is just an example, just so you know it exists. But in the near future, I think we're going to bring some consolidation of all these strategies. Very quickly, I want to talk about what you can do to manipulate and set up the universe, because you can modify the positions of the atoms. Okay, you can do it directly. For instance, you can select a protein like this, and then you can just modify it like this. You assign to the positions. If you assign the positions, they will change. In something like this, for instance, what we're doing, we're subtracting the center of mass of the protein. So essentially, we are just saying that, okay, we're going to center the protein at the origin of our box. Very simple. Okay, 2 lines, it's done. But it can also be a side effect. And so I use side effect in the sense that you run a Python function and it does something to the positions in the background. Okay. For instance, there's this function, pack into box. This one will take any atom that's outside of the box and will use the periodicity rules to put it inside the box. Rotate by, okay, this makes sense. Okay, you're going to rotate the protein by 90 degrees over the z-axis centered on the origin. Okay, there's another one. There's an analysis which is about alignment. So for instance, you can align, in this case, 2 proteins or whatever structures you want. It would go like this. You would load 2 different universes, and you just tell it to align universe 1 to universe 2. In this case, on this carbon alphas, for instance. This is again an example, and the coordinates of universe 1 would have been changed by this. Okay. When do you want to use this? Often as part of an analysis. This aligning is very common if you want to use something as a reference. But also it's very useful to automate system construction and to make it systematic. And this is very important because when I started working with structural modeling, very often I would be using PyMol or VMD, and I would kind of by hand, especially VMD, make it very easy to use the mouse and position stuff kind of where I wanted. But to be impossible to actually redo, if I wanted to redo exactly the same setup, it would be impossible because how can I actually match exactly the same pixel I hit with the mouse? So this makes it very nice to have reproducibility and a sort of a computational lab book that you know exactly what happened and how it happened. Okay, so this is important. All my students are urged to use this approach. There's more building tools. We can merge universe, essentially just add two universes together. So now, or two atoms, two atom groups together, or several atoms groups together into a third universe, something like this. Now they're part of the same universe. We can create it from blank. You just need to know how many atoms you want, and then you have a blank universe. Then you can construct stuff by modifying the positions of these atoms. And you can actually write and at the same time convert the structures. For instance, here we load the protein, we select the alpha carbons, and then we just write them. And in this case, we read in a PDB with all the atoms and wrote out a GRO file from the GROMACS suite with a subset of the file, subset of the atoms. Very simple, 3 lines, it's done. Okay, this is very handy. And the last slide, last relevant slide, visualization. So this, I myself don't use this so much because I have old habits, but my students really found this very useful, especially during the pandemic, that you can use Jupyter notebooks. And if you couple this with NGL View, the plugin, you can load a structure with MDAnalysis, load a universe, and use NGL View to actually interactively view it in the notebook. So this is great because then you do your analysis and you see your results live and you manipulate them, so you don't have to write the structure open in VMD or PyMol or whatever. And so this is super handy, and especially during the pandemic, everyone working remotely, perfect. Our computers at work were running Jupyter. We were just forwarding the connection and working flawlessly. Now, one thing just to finish, and this is sort of the acknowledgement. If you are interested in this, you probably are asking, okay, but how solid is this MDAnalysis project? Because if you're committing to learning something and to writing your code in something, the project better stay around for at least a couple of years. Otherwise, it's going to be a waste of effort, in a couple of years down the line, no one's going to be using it. So actually, the health is pretty good. So I think I'd say that this is a very exciting time for the project. There's a very engaged developer and user base. Everyone's welcome to join. It's you can get in touch using GitHub, Discord, the Google Groups list, mailing list. There's already several downstream projects that are known to use MDAnalysis, so this is great because this creates a sort of a chain of dependency and and feedback, and which is great. This is developed under the NumFocus umbrella and sponsorship. So NumFocus also sponsors stuff like NumPy, SciPy, Matplotlib. So this is great. They recently MDAnalysis got awarded this development grant by the Chan Zuckerberg Initiative. So there's even more money now to code more, which is great. It's frequently a Google Summer of Code participant, which is also great because you bring in more talent. And there have been frequent workshops. The last one was April this year that I also co-organized and sponsored in this case by Surf and Praise. So this is all good signs that the project is alive and kicking and is going to stay around for a long time. And I'm going to finish here. I'm going to thank all the core devs here in, I think this is alphabetical order. Although Oliver, I'd say, is the most involved mentor of the whole core dev, so credit where it's due. Also thanks to the Emeriti developers. I'm part of them now, but the original authors, Jan Domansky for actually introducing me to this, and Michaela and Sebastian. And thank you all for listening. So,
Manuel and Melo.
I can take some questions now, but I think they will have to be read to me from the YouTube channel, right?
Yeah. Can you listen to me? Professor Manuel Melo? I don't think you can hear. Are you listening?
I don't know if you're saying anything, but I really can't hear anything.
Yeah. Professor Manuel, can you hear me? No? All right, I am receiving some information backstage that people can hear me, but not Professor Manuel and Melo. All right. What about now? Can you hear me? No? Oh, it's a pity. There are very interesting questions in the chat, but I don't think that Professor Manuel can hear me. Let's try to figure something out.
Okay. I've seen that there is a problem that I'm apparently not hearing the audio. I'll just quickly refresh this.
All right. All right. So I believe Professor Manuel and Melo had some problems with his connection. Then I just like to advise you all that we soon will have another lecture. Uh, okay, then I'll enjoy to introduce Professor Michael Schertz, and then we try to send the questions directly to Manuel and Melo, and he can answer you all. Okay, so all the questions are going to be sent to Professor Manuel and Melo, and we'll try to maybe you can let your email commented here for those who asked questions, and we can directly send to him. All right, moving on. We have now up next the Professor Michael R. Schertz. I'll go. Good afternoon, Professor Michael R. Schertz. Thank you. We would like to acknowledge your availability to be here with us, to share part of your work with us. Thank you so much for your time. Okay, I'll provide a brief introduction of you, and correct me, please, if I say anything wrong. So, Professor Schertz is an associate professor of chemical and biological engineering at the University of Colorado, Boulder. He was awarded with several prizes, being the most recent one achieved in the last year, which is the AICHE, right? Computational Molecular Science and Engineering Forum Impact Award. He's seated for increasing the utility, power, and reliability of molecular simulation as a chemical engineering tool through development and improvement of statistical mechanics methods and programs. Schertz Groups designs and characterizes new materials at the nanoscale by using theory and computation. Their focuses includes drug design through the prediction of physical properties and binding affinities, and the design of novel biomimetic materials. He has an H index of 43, with more than 16,000 citations. So, Professor Schutz, thank you so much. We are honored to have you here with us. Feel free to start when you're ready. The screen is yours.
All right, thank you. Well, it's a pleasure to be talking to you all. Hopefully this will be something, you'll learn something, it'll be something valuable. It's not quite at the level of tutorial as Professor Melo did, but it's designed to be hopefully pedagogical at the graduate student level. And that's what I try to do, try to make sure that the research is useful for people. And H-index is a silly measure, better the number of citations, but not great. Just focus on making sure your good research gets out there and helps other people in what they do, because we're all building on each other. So let me share my screen. Share screen. And here we go. All right, so I'll be talking about using mixture distributions and maximum likelihood to calculate thermodynamic properties from simulation. So overall outline of this talk, I'll be talking about what we're trying to do when we simulate molecular systems. When you have a molecular simulation and simulate it, what is the goal there? And I'll talk about using reweighting or importance samplings to get more information out of collected ensembles. You worked very hard to get your simulation to simulate something. How can you collect the most information you can out of it? And finally, understanding a little more about potentials of mean force, again, without using histograms and why that's a bad idea. So first, I'm going to do a complete overview of using simulation to calculate thermodynamics in 5 slides. So very, very, very high level, space station high level. So we simulate molecular models to understand how thermodynamic properties change according to our hypotheses. It's always very important to remember that we're doing modeling. You know, we have models of how molecules behave. We shouldn't expect them to be exactly right. No model is exactly right, not even the analytical models that experimentalists might use to interpret the light waves coming off of their experiment. But with molecular modeling, it's tricky because we actually do put in a lot of the details, but not all of them. So I think it's important to always remember that we are essentially testing hypotheses. If you build a model that has properties A, B, and C, and you observe phenomenon X, then you have proven that A, B, and C are sufficient to cause X. That might not be exactly what's happening in experiment, but if you always keep things hypothesis-based, then you've managed to prove something. So we do, just some examples of things. We look at stability of molecular polymorphs, understanding protein peptide binding, self-assembly of liquid crystals, and all of these are described by an ensemble of molecular configurations, which we hope our models capture the features of that true ensemble well enough. So properties of thermodynamic interest are ratios of highly multidimensional integrals over coordinates. Since we're interested in ensemble properties, that gives rise to thermodynamics. So for example, here, it's some observable of the system. The volume of the system, in case you're looking at volumes or densities, a radius of gyration, if you're looking at the size of some macromolecule. So that ensemble average that we're interested in can be, and here, U is, I'm using U to mean beta U. I'm trying to clean up the notations by making it as small numbers and symbols as possible. We have some observable that's a function of the coordinates of the system. Each individual snapshot in our ensemble has some coordinates. And then A is all the parameters of the system. Temperature, pressure, the details of your force field. If you have some coarse-grained model, something to do with the stress on the field, any sort of parameter that's baked into the model. So the thermodynamic averages are functions of the parameters, not the individual coordinates. But it can be expressed as an integral weighted by the Boltzmann factor of whatever simulated ensemble you're interested in, normalized by that Boltzmann factor. So on the top, it's an integral. On the bottom of it's an integral. It's a ratio of 2 integrals. And these are very multidimensional. I remember how painful it was in multivariate calculus, integrating in spherical coordinates over 3 dimensions. That was a gigantic pain. This is over 100,000 dimensions. So there's some very smart analytical theory on statistical mechanics where you can make some simplifications and make these simpler integrals. But fundamentally, thermodynamic properties are ratios of these integrals. Free energy differences are also ratios of integrals. Now it's just the ratio. You have two different states, say they're different temperature or different pressure, or you've changed something about the system. It's a ratio of the Boltzmann factors with one set of states divided by the Boltzmann factors with another set of states. Here, this F, it's some free energy, whether it's a Helmholtz or a Gibbs free energy. It'll depend on what simulation you're doing. But all of your free energy differences are just ratios of two integrals. So if you're interested in thermodynamics, kinetics is another story. That gets a little more complicated. We'll stick with thermodynamics here. If you're interested in thermodynamic properties, what you really want to do is calculate these integrals over all the possible ways for your molecules to be arranged, weighted by things like temperature that can affect the probabilities of each configuration in those ensembles. Nope, this is 4 slides. Let's don't go on. Still 5 slides. Using molecular, so what we do, now that we want to get these integrals, we use molecular simulation methods to perform Monte Carlo integration of these ratios of integrals. So what do I mean? So let's say I have some integral. This is just an integral of each conformation, some observable, integrating the observable at each conformation times the probability of that conformation occurring. What Monte Carlo integration is a technique that you can replace this integral over an infinite number of, not infinite, practically infinite number, to 100,000 variables into a sum over observations. And the key is this will be true in the limit of collecting enough samples if these configurations are sampled from your probability of interest. So we replace an integral that you have to do all sorts of fancy calculus with to ever get an answer with a simple sum over observations, as long as you can pull those observations from the correct distribution. And this will be correct if you can collect enough representative samples. So that is the key. We turn a problem of integration in variables into a problem of sampling configurations. So what we just have to do is come up with some method that samples sampling that generates the Boltzmann ensemble. So for example, molecular dynamics using small collective moves, we apply Newton's equations of motion, solve the coupled PDEs, then as long as we have some way of introducing thermal noise, we generate the Boltzmann ensemble. Different Monte Carlo methods generate the Boltzmann ensemble. So that's what we're doing. We're finding these integrals by using Monte Carlo methods, and whatever the method is, here's the catch. Here's the catch where we take this intractable analytic problem and turn it into a simple set of averages. Whatever method we use to generate for the Boltzmann ensemble, it will almost always have long correlation times. We don't have any good way of picking, of all possible protein configurations or all possible configurations of some alloy, what the right ones are. We have to use methods that move towards the Boltzmann distribution but are very slow. And that's one of the biggest problems in molecular simulations. And nowadays, if you're interested in the properties of a fluid, no problem, you can converge this. But almost anything that's interesting will have very long correlation times. And so that's all the other papers you read on your classical molecular simulation are really about identifying improved ways to accelerate sampling through this multidimensional space of interest. Umbrella sampling, replica exchange, Hamiltonian replica exchange, accelerated MB, multi-canonical simulation, adaptive biasing force, weighted on that. You can keep on going and going. You'll find thousands of papers, maybe not that, at least hundreds, probably thousands of papers proposing new methods. Half the time they're just actually old methods relabeled because they didn't read the old papers enough. It's a gigantic zoo. But that's really all it's about. It's how can we accelerate the rate at which we get these samples from the Boltzmann distribution, different samples that are as different as possible. And then you identify improved ways to estimate multidimensional integrals, giving the molecular simulation data. I've got this precious data. Is there a way I can do the analysis that's a little more complicated than simple averaging, but gives me more information? So free energy perturbation, Redound acceptance ratio, WAM, MBAR, there's a lot of these as well that are better ways to analyze data collected from these simulations. And that's it. That's what we're trying to do. We have hypotheses about molecular models, we collect the ensembles, and then we compute thermodynamic properties from them. And if the system's simple enough, you don't have to go through all the rigmarole. You can just do a very simple sampling. But if not, you need to do a lot of work in order to be able to answer the hypotheses that you have. So that's sort of the overall overview. That's the way my group likes to think about this. A couple advertisements for ways to get more information about these ideas before I dive into this particular topic. Live comms, Living Journal of Computational Molecular Science. Visit those to learn up-to-date molecular simulation techniques. So what it is, it's a peer-reviewed journal for papers that can and should be updated. Like, what are the best practices for calculating protein ligand binding affinities? What are the best practices for computing uncertainty in molecular simulation? Tutorials, high-quality tutorials, computational comparisons between codes, reviews that are updated perpetually rather than having to be like rewritten in a different journal each time. 3 issues published, another issue is partially done, hopefully be published soon. So definitely check these out and get involved in updating and writing these articles. This is sort of an attempt to make the literature work the way it should, as opposed to the way that helps journal publishing companies work. Physical validation is a set of tools to help make sure that your simulations are doing what you think, that they're actually sampling the Boltzmann ensemble. Wide usage of molecular simulation calls for simple to use physical validation tools. So we have some tools that can validate: is your kinetic energy distribution correct? Are you actually sampling from the ensemble you think you are? Is your integrator behaving correctly? So recommend going to physical validation, read the docs.io for more information, and contact me if it's working, it's not working. These are tools that are designed to make your life easier to check and say, oh, wait a minute, this is not just, it might, the average kinetic energy might be consistent with the right temperature, but something's very wrong. It's very there's something wrong with the deals. There's something wrong with this. It's not obeying the laws of physics, so I'm not getting the answers I want to. And finally, there's a YouTube channel where a lot of the talks that we have previously can be checked out. And for example, the testing molecular simulation results. I won't be talking about this really anymore, but you can read a lot more about how to get that to work online. Okay, so now that we have this idea of we do simulations and then we do some sort of Monte Carlo averaging to calculate properties. One trick that we've used a lot is a statistical concept of importance sampling. But that's a very vague term that doesn't necessarily hard to understand exactly what that means. So let's, I'll describe what I mean. So as I said before, if you have some observable, you can write it as an integral times the probability of the chance of that observable occurring at each coordinate. So we take this integral, and what we do the thing that mathematicians always love doing. We multiply by 1, right? But a particular kind of one. So in this case, we multiply and divide by some other probability distribution. What is it? Doesn't matter. As long as it's not 0 anywhere, we can multiply by any probability distribution we want to. And we're multiplying by 1. We haven't changed anything. So now, ooh, tricky. I swap these. I swap this j and this I. All I'm doing is moving this on top of the fraction, moving this down below. So now what we can do is, we can turn this into Monte Carlo integration, where now instead we're interested in properties under state I, temperature I, whatever. But what we do is we sample the configurations from a different probability distribution. And then what we need to do is multiply each of these by this ratio of the probability distribution we want to see divided by the probability distribution we actually simulated. And so what you can do is essentially you're sampling from one distribution and getting observables from another distribution. If this Ix divided by Pj is similar in magnitude as Xn varies, we can avoid generating the entire Monte Carlo chain, i.e. the chain of trajectories that we generated. So that's the basic idea. It seems really powerful. It's fairly powerful. It's not quite as powerful as you might expect, because if these two probability distributions are different, then we don't actually, if they're too different, we don't get that much information, right? You can only average configurations you find. So if the configurations that are interesting of interest in ensemble I never occur in ensemble J, you're not going to get any results. But what can be useful is if the observations are there, but they have the wrong weight. And so you can reweight them and sort of correct it to get better behavior. So I'll show you some of the things that we've done to do this. For physical problems, I talked about a general probability distribution. That's interesting. I haven't said anything about Boltzmann distributions yet. Of course, we usually use them with Boltzmann distributions. We know our probability distribution is proportional to e to the minus beta u. And the normalized probability distribution we can write as that probability divided by the partition function, or this is a way I like to write it because then everything's in the exponential. The probability in state I of configuration X is e to the beta, the free energy of I minus beta, the potential energy of that particular configuration. Free energy is independent of the configuration. The energy obviously is not. So that's the probability distribution we're interested in. And so if we look at this ratio that we need to reweight everything by, it's just beta delta free energy minus beta delta ux. All of these things, this we can, whatever our model is, our model gives us a rule for this. Free energy is a little more complicated. How do we do that? I'll talk about that in a second. So your observables, this is the case of molecular simulation. What you need to do to find out the probabilities, the observations with average I generated using J is you find the energy difference between the two states, take the free energy difference, and you can get that reweighting. Now, one problem is, what about this free energy difference? You don't necessarily know that. Well, what if you plug in that your observable is 1? If you plug in that the observable is 1, you can solve to find that, oh, the free energy difference can just be estimated by the average of the potential energy differences. And so that's your formula for the free energy. There's ones that are more efficient, and then you can plug them in. So essentially plugging it in, this is what you get when you plug everything in. There, the free energies don't appear here, just the energies. So that's the idea of importance sampling and how you can apply that to molecular simulations. Free energy perturbation, you've probably heard of, is a variant of this. This is sort of putting everything, deriving these things from first principles, just from the statistics. Yeah, the thermodynamic expectations, that's the formula. So now a fun thing is we don't have to do importance sampling from just one distribution. What if, say we're interested, we carry out this simulation and we're interested in properties, this probability distribution puts gives us information near coordinate 0. What if we want information at coordinate 2? Well, we could run a simulation there. But what if we want information at coordinate 0.1 or 0.2? Which distribution do we reweight from? Do we reweight from the red one, the blue one? What if we want information about all of these coordinates? Do we have to do a simulation that sort of localizes everything? What we can do is create something called a mixture distribution, which is just say, I like them all. Let's just put them all together in a single pot. And so this is the mixture distribution, all of those. Now we have observations everywhere. This is just normalized by the number of distributions. If you have a different number of samples, the mixture distribution looks like this. You just multiply each of these distributions you sampled from by the fraction of the samples that occur from that. So you can do importance sampling from multiple probability distributions. It's called mixture distribution. You're mixing it all together. Yes. So you might have heard of something called MBAR, multi-state Bennett acceptance ratio, a cumbersome name that I came up with a while back. And if you've read that original paper, it's really complicated. At the time, I didn't know enough to make it simpler. But since then, I've come to understand that there's a much better way to understand what this is that the math seems complicated, but what it really is a way to find free energies and observations that are sampling from the mixture distribution. If you have multiple simulations carried out at multiple states, then this is the formula you get from that very complicated paper. But you can notice that the denominator here is really just the mixture distribution. That what we're doing is we're finding the free energies of each of the states. Let's say there's k different states, by reweighting each of those states from this mixture distribution. It turns out that these are dependent equations. So if you have 3 states, there's 3 equations that you have to solve simultaneously. There's actually only 2 because the reference free energy is you can set to be whatever you want. So what we're doing, this MBAR, is important, collecting data, turning that into one big distribution, and reweighting from that distribution. So if we have samples everywhere we care about, we can figure out the correct way to average them. If you change your temperature a little bit, or change an alchemical parameter a little bit. So yeah, this was published way back when. Reduces to Bennett acceptance ratio for 2 states. Weighted histogram analysis method in the limit of 0 width histogram bins. I'll talk about histograms more a bit. You can think of the maximum likelihood estimate of all the data collected. Of all the data you collect, what is the most likely estimate of the free energy differences between those states? You can prove that it's the lowest possible variance reweighting estimator that's also asymptotically correct, meaning correct if you collect enough data. PyMBar is an efficient Python implementation that we've developed. I'd encourage you to check that out. We hope to get 4.0 out over the holiday break. We'll see how that goes, but there's pretty good versions up there already. Whoops, wrong. Can you still hear me? Okay, yes. All right. Yeah, so if you're interested about PyMBar as mixture distribution, there's an archive preprint that was published a few years back that talks a little more about how to think of it that way. So great, we can do important sample. We can collect several simulations and predict a lot of properties just using a few of those simulations. But what are the useful distributions that we can sample from? So we talked about calculating properties of models. We look at the properties, what the reference distributions are, and then the target distributions, what we're trying to predict. So let's say a temperature scan. We have a few reference distributions, and then we can construct the target distribution over many things. Like, for example, we're interested sometimes in calculating heat capacity curves of peptide folding. And so we do a few simulations and then put those together, usually like a replica exchange in temperature, and then we can get a continuous probability, essentially continuous plot of the heat capacity versus temperature using this. Free energies of binding, this is very common. You're interested in the free energy of binding some ligand, and you have your coupling parameters. Your target distribution is still the same parameters, but as long as there's overlap between them, you're using essentially all the information from all the states you collect to predict the free energies. Potentials of mean force, you have some reference distribution. You like localize the probability at specific values of some collective variable. And then your target distribution is some what the unbiased, what the distribution, what the free energy is without the umbrellas that you put in. Parameter scans. If you have some model, you have a bunch of different force fields parameters. You can do some scan of how do the properties change locally as I change these parameters, without actually doing those simulations. So one thing I want to compare to is the weighted histogram analysis method and why this approach is better. One problem is that these properties are often computed by histograms, where you, rather than collect the values, you bin the values together and then solve a set of self-consistent equations. So this is one version of the self-consistent equations of WHAM. And so let me explain what all these are. So the index I labels the histogram bins. So you have 10 bins that you bin all your information in. The K labels the number of biased states. Say you have K different temperatures or K different harmonic restraints that keep the simulation within a specified range. And then N is the number of counts per bin. And PI labels the probability distribution of bin I. So this is a probability of a bin. FK labels the free energy difference of the biased states. So you have two different things to keep track of, the bins and then the states. And those are two different numbers. Now, one problem with these equations, is that they assume the same biasing potential everywhere in the bin. But if your bins are too big, then that's an approximation. And you don't want to include approximations if you don't have to. Another issue is that this PI assumes that the same probability everywhere in the bin. But we know that most things we're interested in are going to have some continuous distribution. They're not just necessarily the same everywhere in a bin. Things are continuous, not discrete and lumped into bins in most cases. Unless you're studying Ising models or other discrete models, in which case, yes, they are. And as long as your bin is that width, you're fine. So other problems, for example, in multiple dimensions, the number of histogram bins required scales with your resolution to the dimension, which scales pretty badly. So although it's used, and if you choose your bin size correctly, it reduces to M bar. They're M bar. There's still some issues. So what's the fundamental difference? So one way to think about this is looking at bitting and energy. So in WEM, when you use WEM on, say, energy states you get with, say, replica exchange and temperature, what you're computing is a density of states. And density of states are very useful. A lot of thermodynamics can be expressed in terms of density of states, can be used to compute properties. An average is the observable is a function of energy times the density of states times the Boltzmann. That works great. What M bar is, instead of representing densities of states as histograms, it represents this as delta functions. The probability density is a weighted sum of delta functions. And so your probability distribution, it's not really a function, it's a mathematical object called a distribution, but it behaves mostly the same in most cases. It's a sum of delta functions where you collected that data, and the weights change depending on, say, if I collected data at 300 degrees and I'm interested in how it behaves at 300 degrees, all my weights are 1 over n. If I collected at 300 degrees and I'm interested in the properties at 290 degrees, then the weights are bigger for the lower energy ones and lower for the higher energy ones. And so those weights are what change when you try to predict properties at different states. Not good for visualizing. Very hard to visualize what this means. It's very good for computing integrals in high dimensions. Because if you remember from calculus, the integral of some function times a delta function is the value of that function. We're turning integrals into sums, which is what we're trying to do with the simulation. So now instead of this complicated expression, we just have a weighted sum. Histograms can result in significant bias. Here's one study where some people were calculating probability distribution of a molecule dihedral angle. And if you choose your bins wrong, WAM can give you very biased results, especially for the barriers. And the barriers, the gray is the true result. The green is what WAM gives you, whereas M-bar gives you the correct occupancy of each of these bins because it's not making the same approximations. So I would say it's provably like the best reweighting method to do it. Sometimes it's only marginally more great. For example, if you're looking at free energy calculations, we did a bunch of benchmark tests a while back, and we actually repeated the calculations 100 times. Like a lot of times you talk about, okay, we'll repeat the calculations a few times to get uncertainties. We repeated these calculations 100 times to actually compare analytical uncertainties with the sample variance. 10 different free energy methods, and you can see here, these are actually not the errors. These are the errors in the errors. So the green and yellow are the errors. The error bars there are the uncertainty in the errors. And there's a bunch of methods. You can see that, for example, M-bar, thermodynamic integration, if you have enough lambdas, ends up working as well as M-bar. Bar works about as well as long as you don't trust the uncertainty estimates of bar for some subtle reasons that we won't go into here, having to do with correlated samples. But other free like a simple insertion or deletion free energy does not work as well. So if you're very careful, you can do as good as M-bar. It's a limiting case in many cases, but just something. It's an approach. So if you're doing free energy calculations, you don't have an implementation of M-bar and you have bar. As long as your spacing is good, don't worry, you're doing great. So a couple fun things we did: use reweighting to rapidly validate simulation parameters for free energy. So this is sort of a fun test. Usually when you're doing simulations, you have some cutoff. You transition from short-range interactions to long-range interactions at a cutoff. So where should your cutoff be? Well, if you want to do a free energy calculation at each of these cutoffs, that's a huge number of free energy calculations you have to do. And it's especially hard because you know that the answer is going to be pretty close. So if you have some number and you're that's noisy and subtracting another number that's noisy, the answer is going to be noise. You're not going to get much information out. So what we did is we looked at just carrying out one simulation, changing the cutoff, reevaluating the energies, and seeing what we could learn about the free energy differences. And so this is sort of a cool thing. And we looked at 5 different variables, and you can see as we change the variables. So this is all generated from one set of data. So we just did the alchemical transformation once, and we looked at how the data behaved. So what we're able to do is identify simulation parameter choices with negligible difference from expensive calculations. So here there's sort of 3 free energy differences we're interested. our initial simulated set, the ones with, a reasonable cutoff, the expensive parameters, we make all the cutoffs so as big as possible, and optimized parameters that are very close to expensive parameters, but are much cheaper. So what we're doing initial state sampling, and then calculating these free energy differences, but also calculating this free energy difference between simulations we never did. So this is for anthracene solvation. So we're actually making something pretty big and sticking it in and out. So one thing to pay a look at here is this column. Let's see. So these are the predictions using just the benchmark set. You can see that the benchmark ones were, quite different, about 0.6 kilojoules per mole away. You'd rather have less there than that. The direct free energy differences is very hard. If we're trying to predict a 0.6 difference and our answer has about 0.17 error, it's very hard to tell if we're doing better or not. You can't really tell. And so here are, these are the predicted, here we figured out what the optimal parameters were without ever doing the simulations. And we got the answer right to 0.03 kilocalories per mole. When we actually did the simulations at all the states, the answer was almost exactly the same. So basically, we're able to push the uncertainty. Because these states are relatively similar to each other, we can explore the properties of all the similar states only doing one simulation. And so that I think that the key is the fact that this prediction, remember, only sampling from this state, was essentially exactly right, and we were able to identify what the best simulation parameters would be. One key to all of this: how can we get an answer that's so accurate when everything is so noisy? And that's a key of this. If you look at the variance between two experiments, so these two quantities we're calculating, you can expand that and break that down. Is that the uncertainty is the sum of the uncertainty? So the square variance is the squared uncertainty. You have the sum of the squared uncertainties. And usually you stop there because usually you have completely independent results. But in this case, we don't have independent results. We know that obviously if we're calculating this free energy with this exact same configurations, then it's actually highly correlated. It's extremely correlated. The great thing about MBAR is it actually gives you this covariance estimate. So you're able to find very small uncertainties because you know the variance and you know the covariances between the two. So rather than getting very large numbers and uncertainties, we get very small numbers and uncertainties. That's the key. So it can be used to fill out a multidimensional thermodynamic space. This is some work from Charles Abreu and Ed McGinn, where they actually used MBAR to fill out a multidimensional phase space with carrying out just a few simulations. So they had a nice way to phrase it, that it could be seen as a numerical equation of state, that you fit, you do a few simulations and then you fit everything else between, but you're not just interpolating, you're using a rigorous statistical framework to do that. So PyMBAR is a Python implementation of reweighting, fast, robust, efficient. There's lots of examples you can use. Potentials of mean force, salvation for energies, forced by a single molecule experiments, actually used to analyze experiments. Automated setup.py installation through Conda. And so it's hosted the GitHub of one of my collaborator on this, John Cadera. And so I'd encourage you to test it out and let us know how it works, how it's working for you. And yeah, it's been downloaded hundreds of times, and we respond to the bug reports. And yeah, so try it out. Oh yeah, one of the cool things we did with this was to scan molecular simulation. Here, let's look at some science, some testing, some models. So let's say we're interested in Leonard-Jonesium solvated in water, like single Leonard-Jones particle in water. So there's two parameters, sigma and epsilon. What is the free energy as a function of these parameters? So let's start with 10 molecules linearly in volume space, and then predict the salvation for energy along a 2D grid of states. So this is what we did. So we just had to do these free energies of salvation and then used reweighting to calculate the properties at all the other states. This is one of the most, this is so fun to do. We have here the free energies. You can see the free energy is a function of the size of your Leonard-Jones sphere. And here's the uncertainties in these. So you can see in this range, we did a great job, but it turns out that we really didn't sample low sigma well. So we threw in one more state and we drove the uncertainty down much lower. So this really, in just 13 simulations, we've essentially, or I guess it was, we did these two, yes. So which one? Oh, actually, no, we did 11, that's right. And so we're able to capture essentially, physically representing things from methane to neopentane to, if you're modeling your buckyball as a Leonard-Jones sphere, capture all of those with very high uncertainty using this framework. So, 28,000 salvation free energies in about 1 hour in one CPU, rather than doing all those simulations. So what was even more fun was looking at free energies as a function of 3 parameters. What about ion salvation? If you're limited to Lennard-Jones, then essentially you've got 3 parameters: your charge, your sigma, and your epsilon. And so this process took about 16 CPU hours for simulation, 25 seconds to compute potentials at the other states, and 5 minutes to 7 hours to compute free energies, depending on how things were set up. And so using some clever adaptive techniques with about 200 states, then we're able to essentially go through and calculate free energies as a function of all these 3 parameters, with a huge scan from Q plus 2 to Q minus 2 all the way. So why is that relevant? Why should I care you can do that fancy pants calculation? Well, we talked about doing sampling in order to test theories. We can test salvation for energy to analytical theories with unprecedented levels of detail. For example, we can look at the difference between explicit salvation and boron salvation theory. You can look at things like the asymmetry in salvation of charge in water. Here are the free energies as a function of sigma and epsilon at positive and negative, 2 values of positive and negative Q. You can see that the free energies are quite different depending on if it's positive or negative, and that's not included in boron salvation theory at all. So this is something I wish we had followed up on more, because if anyone wants to analyze these data sets, we can still dig them out. It's fun stuff. And I think I'll talk in a few minutes a little bit more about how reweighting can help clarify what it means to compute a free energy surface or a potential of mean force. So for a lot of theories, like different rate theories, involve calculating free energy barriers. And to do that, you need to calculate the free energy as a function of some reaction coordinate. So analytically, what this is defined as, I want sort of the expectation value of some set of collective variables, like the distance between 2 molecules, say, evaluated at that expectation. So in other words, the free energy constrained to just something like, what's the free energy if these 2 particles are this distance from each other? So for finite sampling, traditionally this delta is replaced by an indicator function, where that indicator function could be a histogram, a Gaussian, some sort of triangle. And then you perform Monte Carlo integration, same idea, to calculate the expectation value of this indicator function. What's the expectation value that my 2 particles are between 0.5 and 0.75 angstroms from each other? So this could be any number type of indicator functions. And then one way to do that, one problem with this is, of course, that if you're interested in barriers, you get no data in the mountain regions, right? Because the simulation doesn't want to hang out in that area. It wants to hang out in the valleys. So one solution is you can collect data with bias simulations, so we get samples everywhere we care about. So we determine the weights from K umbrella functions, and then we construct the free energy surface. We can have any number of indicator functions. So we have weighted samples, but by constraining it with some harmonic biasing potential, and then we have the indicator functions that we say, okay, how likely is it to be at different states? Now the thing is these really should be 2 separate steps, but they're almost never treated as separate steps. Where you have essentially the bins that use your indicator functions and your umbrellas are frequently almost always treated as the same thing. And so you have the same problem with free energy surface that it does with other properties. When we talked about before, confusion between the biasing potential being the same everywhere and the probability being everywhere. So so essentially you get, if you're looking at potentials of mean force for some alanine bipeptide or whatever, then you get, if you use if you have enough bins, then it behaves correctly. You get the same answers with M-bar and WAM. But if you use different numbers of bins, these get offset. So the key is that the free energies should be determined for each umbrella simulation, and the weights should be determined for each sample rather than being mixed up. So I talked about this before. So a better way to think about the problem: what do we actually collect when we simulate something? So let's say we have some collective variable, and we're trying to identify where this barrier is so we can calculate rates or something. So we know that underlying the whole problem is something that looks like this. We have some collective variable, like the distance 2 particles are from each other, or going along some dihedral. And there's some barrier there, and it's usually pretty smooth. But what we actually get from a simulation is this. We get lots of samples where there's high probability and fewer samples where there's low probability. But we get this set of samples rather than this smooth curve. And we could do something like what's called a kernel. We just like put a little Gaussian down, kernel approximation at each point. And then that sort of looks like a smooth surface, but it's not that smooth. It's a little unsatisfactory. It'd be a hard time like actually calculating transition rates over that. You make a wider distribution. It looks a little smoother, but clearly, if I use a wider Gaussian, then they stop looking quite alike. So what happens when we perform a simulation with biases? We know that when we bias it by nailing it here, I've got a harmonic bias of 0.1. Here I've got a harmonic bias of 0.65 to force it to go over there. These are what the distributions should be, but what we get are these distributions. So of course, this idea for mixture distribution, if we want to understand what's happening everywhere, we're going to combine these. We find the weights of each sample using M-bar, and we get something like this coming at it. We combine these three simulations with the proper weighting, we get something like this. And this is, I always thought was really cool, because what we have is the information is represented in two ways. There's both spacing information and information about the heights there. So you've encoded what the underlying probability distribution is. It's still a bunch of delta functions, but it's weighted delta functions from each of the underlying simulations. So this is what we call our empirical distribution. This is the data we actually collected. We never actually got any information about a continuous distribution. This is the data we got. And if we're going to have to guess what our underlying distribution is, it should look like this. So how do we generate a continuous distribution close to this thing, right? What we want is, what is the continuous distribution that most resembles our empirical distribution? And so it turns out statisticians have some good ways to calculate this. It's something called the Kullback-Leibler divergence. So this QX can't be an empirical distribution, because then you're dividing by delta functions all over. But P of X can. So what we can do is we can calculate, and in this case, the integral turns into a sum. Yay, integrals turning into sums again. And we can plug our empirical distribution, i.e. this set of delta functions, and some other distribution, some trial distribution, and we can see how close each trial distribution is to our sets of delta functions. So what we want to do is find the probability distribution that has the smallest distance from the empirical distribution. So now it's an optimization problem in the parameters of our trial distribution. What do we represent this trial distribution by? Well, really, it depends on us. We have to decide. Anything we feel it's reasonable should be smooth. We think the underlying free energy as a function of collective variable is smooth. Not too many parameters, because then it's hard to optimize and we don't want to overfit. Histogram bins, well, parameters bin width, but even something like kernel density estimates is better. But even better is something like, say, Gaussian mixture models. One thing we've used are splines, where the parameters are simply the values at knots. This is similar to the variational free energy perturbation work from Lee and York, which was inspired by and closely related to this work. So here's several different cases, looking at umbrella sampling of a valine dihedral in lysozyme L96A. So we have 26 umbrellas with, this is published last year, 26 umbrellas with varying spring constants, 500 data points per umbrella. And we use different cubic splines. And uncertainties here for bootstraps, you can see a few different ways to combine. There's a few different variants here. But you can see that you can get a very good agreement with this. There's a maximum likelihood of interpretation if you don't like the Kullback-Leibler divergence. But basically, this is a likelihood of a given order parameter given the data, which is just a product of what's the probability of observing the points that we've measured. This empirical distribution, what's the probability that this empirical distribution came from a given trial distribution? And so what we just want to do is maximize the chance that of all the trial distributions, maximize the chance that that trial distribution did indeed give all these delta functions that we observed. So how do these methods relate? I won't go through all the math, but basically maximizing log likelihood is the same as minimizing the KL divergence. How do I know how many parameters to use to avoid overfitting? The great thing is now we've turned this into a maximum likelihood problem. There's a lot of different ways to look at error fitting. For example, Akake or the Bayesian information criteria. Here what I'm looking is the information criteria is a function of the number of splines. And what you can see is that you really need about 14 or 16 splines, depending on the criteria you're using. It's interesting how this behaves. Let's stick with we want to look at the bias states version. The red line that's not relevant for this. The blue line is the relevant for the one where I'll be graphing. And you can see that this basically tells you around 14 to 16 data points is the number you want. And yet, indeed, if we look at the PMFs at say a 10 is the red line, it underfits. And if you look at 30, it's not as obvious, but it's overfitting, and you can show some other measures that really once you get up to 14, it behaves quite well. So 10 is not enough, 14 is, and standard maximum likelihood information criteria will tell you that. So that says that it's a smooth parameter. You need, if it's a spline, you need about 10 parameters to do it. Yeah, I won't go. That's some variants that we're running out of time for. If there's a maximum likelihood interpretation, there's a Bayesian interpretation as well. So you can say, what's the probability of my trial function as a function of the data? And here, then you can explain, here is the probability of the different parameters. That's essentially a prior that allows us to encode things like smoothness in our model. It allows us to say, okay, basically, because we said at the beginning, right, that, or the beginning of this section, that in order to say whether something is, that we want it to be a smooth function, but how do we know it's a smooth function? Like, where does that come from? That's a prior. And Bayesian inference allows you to insert explicit understanding of that prior in. And you can say, okay, given my understanding of what this should look like, and given the data I observed, what is the best prediction for this free energy? Yeah, so what you can do is essentially look at KL divergence, and this sum of Albiant state we think is a better one to use now. And then you can see that this method is not so great because it gives you very high uncertainty in the barriers. You don't want that. You want data more evenly spread everywhere throughout. So here you can see that the uncertainties are quite good. So you get these very good uncertainties that are uncertainties based over the parameters of the model. Yeah, so in this case, we have a prior, we have different levels of prior, and they're basically to favor smooth curves. The smooth line is the maximum likelihood, and then the 95% confidence interval from potential mean in the free energy surface is the shaded area. We're trying to do this with Gaussian mixture models. This is a collaboration with Annie Westerlund. This is still, we're still making this fast up. It's working. We're just trying to make it fast enough to be useful. But Gaussian mixture models are very promising, and they scale really well to higher dimensions, unlike sprites. So we're still testing that out. So a summary of all this. So multi-state weighting is integrate, right? Bad pun that works okay in English. That really what we're doing, multi-state weighting, is just a way to do Monte Carlo integration in a way that it uses all the data you looked at. It's a powerful formalism for calculating lots of quantities more efficiently. Thinking about M-bar, it hurt my brain for a long time. It took me a long time to figure out the equations and to prove they were right. But once I started thinking about it as mixture distribution, everything made much more sense. It's important sampling from the mixture distribution of all the data you've collected. There's good Python tools to carry out the calculations, and you should think carefully about what one is doing when calculating potentials of mean force using reweighting methods. It's important to think of what does it really mean when you're calculating. If you think hard enough about what it really is that you're calculating, it helps you understand how to better get that information out. Yeah, so for example, we should be looking at continuous potentials of mean force rather than histograms, which have both computational problems and then representational problems as well. So acknowledgements, people working on this, alumni on this project, Levi Naden, who's now at MolC, Himanshu Paliwal, who's now professor at IIT Ropar in India. And this is my group just pre-COVID, since we're all without masks. Many of these people have left now, but Pascal Merce did a lot of the physical validation work. And collaborators, and thanks to NSF for various NSF projects for funding this over the years. And that is what I have. So I'd be happy to answer any questions. So let's see, I see some questions in the chat. Let me scroll down. So could you comment about the challenge to get the salvation free energies from system where ergodicity is difficult to be reached, like a polymer, for example? that's really the challenge in sort of classical molecular simulation nowadays. And that was that 0.4 on the zoo of methods. It's really all about trying to obtain that distribution. Something like a polymer is very complicated, because if you have a polymer in sort of a gel phase or below the glass transition temperature, almost certainly it's not in the Boltzmann distribution. It has some other distribution. So I think that's a real sort of untapped area of statistical mechanics. To what extent can we extract the deviations from the Boltzmann ensemble and use those probability distributions to calculate averages? So I would say, like, the challenge to get any free energies when the ergodicity is difficult, that is the challenge of classical molecular simulation. That's essentially what's left. How do we calculate those averages well? And with the caveat that with something like polymer below the glass transition temperature, it's not actually the Boltzmann distribution, it's something else. And I think there's the general theory of non-equilibrium statistical mechanics is very interesting. Can you figure out the time-dependent probability distribution? That's the real question. Okay. About the mixture distribution approach and sampling on the molecular simulation, can you comment how machine learning can help? Machine learning is an excellent question. So, and to some extent, I think where machine learning is useful is where you have an unknown distribution. And the issue with the Boltzmann sampling is, like, we know what the distributions are. Machine learning hasn't been very useful there. I think, for example, the potentials of mean force, machine learning can be useful in terms of, you have an unknown distribution that satisfies these criteria. Well, that could be a machine learned distribution, right? Machine learning is very broad, and so one has to be very careful. In some sense, doing optimizations to Gaussian mixture modelings is a part of machine learning. You've got some data and you're trying to find some function that fits it. It's not neural nets. Neural nets have a few too many free parameters in many cases. Some people have used neural net potentials, but I think it's a lot of times you use a little bit of flexibility in terms of making sure you have enough data. Having simpler models can be better. So I think there's a lot of places for machine learning in this. For example, approximating potentials using machine learning potentials that can, you get something closer to DFT or quantum level accuracy with less simulation. For example, which collective variable should I use? I didn't talk anything about which collective variable I could use, and there's some very powerful machine learning techniques, or they appear powerful. We're still working on getting them to work correctly. And when I say we, I mean the field, not me necessarily. That can be very useful in being able to decide what your collective variables should be. And in that case, a lot of times you don't care what the collective variables are as long as they're decent. They don't have to be perfect, they just have to be decent. And so machine learning is great for that, because there's no underlying theory that one has to satisfy. It just has to be better. And machine learning is very good at making things better. So I think that, it's a big good question. I've rambled a little bit, but there's lots of database ways to explore things, and could talk with people about more particular examples there. I think that you should use the laws of physics where they exist. Sort of a general principle of machine learning is, I think of machine learning and physics is, don't try to learn something you already know. Try to learn things you don't know. Let's see. Do we really require the TD-DFT calculation for calculating the spectra of iron? I'm not sure how that's relevant. I'm not interested in IR spectra. I'm not sure where that was. Hi, Andrew. There have been many papers published where authors attempt to optimize schemes to predict partial charges. Could MBAR be used to optimize the process? Yeah, I think it could. Anything where you're scanning over many similar situations quickly, it's very good for that. Many similar simulations, and you want to do lots of them. This saves you some simulation schemes. So I definitely think that could be the case. We can talk about particular examples, and I could give pointers on that. Let's see. Yeah, I think that's the basic principle is, do the things that you're trying to optimize over, are they similar? Yeah. So let's see. Let's see. I try to see if there's any other questions, and that's everything. That looks like everything.
Professor Michael Schurtz. Actually, there are a lot of compliments here. Can you listen to me?
I can hear you. Yes, I can hear you.
Yeah. That's fine. So there are a lot of compliments here. I just would like to comment about the actually a commentary of Professor Luciano. He asked me for me to give a special thanks here live to Professor Andrew Paluk for inviting you to be here, and very special thank for you to be here to share your knowledge with us. I'd like to thank you so much for your very inspiring, outstanding, and passionate talk. That was very nice. Actually,
Thank you.
actually, there is more 2 questions here. Would you mind to answer?
So I'm seeing some questions. They don't look like they have anything to do with this talk. If they just appeared, if they're coming from Mateus. Any comment on organic, inorganic interface modeling for charge transfer? This has nothing to do with my. Is that's going to the wrong channel? I guess I don't know.
I don't know. I don't know. But I actually
I know nothing about organic interface 2D material. Yeah, it's relevant.
Yeah,
so I must be the wrong place. Yeah.
That's out of the range. I actually have one question for you. As I read that you work with simulating binding affinities, right? And I have already worked with another methodology, which is molecular docking. And I would like you to talk about the reliability in the calculation of the binding affinities through molecular docking, because it's a tough topic, right?
Yeah. So my general approach is I want all of my uncertainty to be in the model. I want the approximations to be in the model, and I want my calculations to be essentially exact. So I can say, yes, this, as I change the parameters of my model, this is how the property, like binding affinity, changes. And I feel approaches like docking don't allow me to do that because there's approximations in the integral. Essentially, you're replacing a large number of configurations with a single configuration, or 5 or 6 configurations, depending on how you do it. And there's a lot of the thing is, I haven't spent a lot of time studying it over the last 10 years. I know that talking to people in industry, there was a big study from GlaxoSmithKline, but this is 2008. It's quite a while ago now. But they basically showed that for the docking existed at that time, it was actually relatively good at identifying locations and binding poses for drugs. I say really good. It wasn't 100% by any case, but there's a good chance that if you docked a lot of times and docked over a couple programs and you got consistent hits, there's a high probability that it was putting it in the right place. But the affinity predictions that came out of the docking were generally not correlated with the binding affinity at all. So docking, it definitely is exposing real physical principles, and in many cases, it can properly identify that small molecules are lining up in the right places and identifying, there's a perfect place for this to fit right here. But they're because of the approximations that are made, they're just not that quantitative. So that's I have not studied that. That's my opinion, but maybe a better way to. So I did the 2 ways. Let's look at a practical way. What's happening in industry? Industry has used docking, and it's been useful for them, and they're continuing to use docking. For a long time, they wanted to do free energy calculations, and they weren't efficient enough to use. Now it's computationally possible to do this, and now they're using both of those things. They're using docking to screen large number of molecules, and when they've got it down to a few hundred or a few dozen, they do free energy calculations, and then they synthesize those. So it's really, so from the practical point, is it useful? Absolutely. Drug companies use it. Accurate binding free energy calculations now are sort of the next point in that pipeline. From a scientific point of view, like I said, the way I like to approach it is if I'm testing hypotheses on models, I want to make sure that the thermodynamic properties, given the simplified assumptions that I make about my model, the thermodynamic properties are as accurate as possible, given the uncertainties and given my model. And for that I prefer to use techniques that, if you collect enough simulations, will converge to the correct answer for the model.
Yeah, for sure. Well, thank you so much for your explanations.
Hopefully
there are more comments coming up here in the chat. Everyone is thanking you so much for the presence here.
My goal is to help. I want to do research that there's multiple ways to do research that help people. If you're doing something that's very, very applied, you can go and invent lithium-ion batteries and help a lot of people. But when you're a level back in theory, then one of the best ways to help people is to make sure the concepts and the tools are as useful as possible, so that as many people as possible can then use those tools in their own research to make an impact.
Thank you.
That is what I try to do. So any feedback to help me do that is great.
That's very inspiring, really. Thank you so much for your presence here, for your lecture. It was amazing.
Thank you so much.
Well, guys, now we're closing the second day of scientific activities at the second CompChem. I hope you have enjoyed the set of interesting discussions we brought to you today. We are going to be tomorrow in the morning, 11 p.m. Brazil time, and the link will be sent to your email. Thank you again for the attendance, and see you tomorrow. Bye-bye.